Grazie. So thanks for giving me the opportunity to tell a little bit what we do at my company in terms of artificial intelligence. And happy to see still people showing up despite being Johannes tomorrow. So yeah, this is not going to be a lecture about methodologies in AI or anything like that. I'm sure you guys are better at that than me. But what I will tell is a bit of the story behind our company and how we have started working, at least with AI, to solve some of the problems that we have. So as the title says here, what we are trying to do is make genetic testing become mainstream in healthcare. So until today, this is still a little bit of a boutique type of analysis. You don't always do this type of test that we do. It's typically at the end when the clinician have tried a lot of other methods and a lot of other tests, maybe sometimes going on for years before they finally decide, okay, we may need to do a genetic test. And the reason for this has been, until now, basically pricing, which has been a bit prohibitive. It used to be a very expensive type of analysis. It's not so any longer. But there are other challenges in this. So that's what this talk is about. So what is a genetic disease to begin with? Well, it's something that is caused by an abnormality or a difference that we have in our DNA code compared to what the general population have there. And this can either be that you have it already at birth, and then it's an inherited disorder, but some of these changes are also occurring spontaneously. For example, in cancers, if you're exposed to some unhealthy chemical for a long period of time or so forth. But the tests that we do, they are aiming at identifying the inherited disorders here. It can also be that these changes are affecting one single gene in our whole DNA code, or sometimes there are more complex behaviors, so mutations that occur in several genes, and then we call them multigenic. And to complicate matters, it's also so that the way these act, or the change, when it has an effect and causes a disease, it can do that either in a recessive manner or in a dominant manner. Meaning, because we have two copies of the DNA code coming from both our parents, if it's a recessive gene pattern, then it's enough. It's not enough to have only one change in the DNA. So if you're inherited this, it means that both your parents must be carrying this particular DNA defect for you to get the disease. But then there are also these dominant inheritance pattern genes where it's enough to inherit one copy, only from your father or from your mother. And our tests are able to identify both of these. And the whole concept behind genetic testing, or the whole process behind, is actually quite a lot like finding a needle in a haystack, because in total in our DNA, we have information about more than 20,000 genes in there. And typically in these disorders, it's only one or two or possibly three of these that are affected. So we have to somehow scan through all of these. And actually, we know already by scientific studies out there that only about 5,000 or so of these are actually today associated to any particular disease. So we can already narrow down these 20,000 into 5,000 or something that are actually relevant. We have then developed specific panels where we are testing a subset of these genes, and certain gene groups are associated to specific disease types. So when a clinician is ordering a test, we don't test all the 5,000 that are known to be associated to any disorder, but rather the subset that specifically is known to be associated to that particular disorder. And ultimately, we hope to be able to identify there on the top the single genetic cause of this disease. So I was mentioning before that until now, this has really been quite an expensive business. But a few years ago, there was a technology that made it possible basically to break the barrier and start this revolution that we are currently a part of in healthcare. And what I'm referring to is something called next-generation sequencing. So what we have on the y-axis there is essentially costs in dollars for actually sequencing the DNA of an individual. And up to a few years ago, this was totally prohibitive. I mean, you could only do it if you had a huge amount of research funds backing your research group to be able to do this for one single individual. You were spending millions to do this. But then thanks to this next generation sequencing technology, there was really a disruption because it introduced a totally different way of doing this, where suddenly you could start to consider doing this in some kind of scale. And where we're heading today, or in at least the coming years, is that we will be able to scan a complete genome of a human being for about $1,000 or €1,000. And that's then the same price level as a normal blood test that you take in the clinic, basically. So we didn't invent this, but we have been leveraging this technology in our laboratory. So the process that we carry out at my company is pretty simple, at least when you lay it out here in a schema. So the customers are the doctors that meet the patients in a hospital. The patient come there with a heart problem. They feel very tired. When they exercise, they notice that there are difficulties with the rhythm of the heart and they cannot breathe. Okay, the doctor does some preliminary test and then realizes that, yeah, well, this could be an inherited disorder. So they go to our website, place an order for a test in that particular cardiology category. They take a blood sample from the patient and send it to us. We have a laboratory here in Helsinki. We extract the DNA from that blood sample. Then we put it through our laboratory process. The little thing you see in the middle on the right panel there, that's supposed to represent these sequencing instruments. So they're not very big. You can actually, well, it's about the size of this unit here, where you can put up to 20, 30 DNA samples from different people. You basically push a button. It keeps on doing a bunch of things, pumping in reagents and taking them out, and then taking a lot of images of what it's seeing there. And after about 48 hours, it spits out 0.5 to 1 terabyte of data with DNA sequence information of these patients. We then pipe this through our data analysis pipeline, which is not really pictured here. These results that spits out are analyzed by a geneticist and interpreted, and ultimately we write a report that goes back to the clinician. And then he meets the patient and tells them what they need to do in terms of treatment, whether other family members would need to be tested or not, and things like that. So pretty straightforward in that sense, but there is a lot going on the laboratory side and the interpretation side and the number crunching side. Yeah, this is a bit kind of marketing material here, just telling about that we started out 5 years ago here with a business, built the laboratory and built the numerical part or the number crunching parts of our process. And in 2013, something we started with just one medical discipline, so just a very small subset of tests. But our goal was all the time to become a one-stop shop, meaning that no matter what kind of genetic disorder, we have the test for it. So people would come only to our webpage and they would then look for the appropriate test there. In 2016, we launched 20 medical categories, more than 400 tests covering these. And as of today, yeah, this slide is even getting a bit old. We have analyzed more than 13,000 samples in our lab here in Helsinki. And we have increased the productivity, so we can now push out 3 times as many reports per day and person as we used to do, thanks to some software that we have developed. But we need to improve this even further. And we have, as it says there, also now customers. Actually, we started out in Finland, but we now receive samples from more than 30 countries around the world. And this is if we look just at the samples that we receive per month. And we can see that basically in 2016, there was this steep incline, and that's when we launched tests into all these other categories, not just the one where we started off with. And this is continuing now this year. So the growth is really high, and that's very good news for us. But unfortunately, we have discovered or realized that though we can easily scale some part of our process, some have not been so easy to do that with. So these are just some criteria that we have realized we need to meet in order to move into this mainstream healthcare. And first there was, of course, the criteria or requirements from the field on the left there, that they felt that until a certain point, the accuracy of these tests hadn't been very good, and they were too expensive, and it took a lot of time to deliver the results. And when I say a lot of time, until a couple of years ago, it could take about up to 6 months to perform a test like this. And we introduced 21-day turnaround time in this. And at that point, when we did that, that was totally revolutionary. Another aspect of things was that they felt the results or the reports that were given by other companies at the time, they were just not fulfilling enough for the clinician, for the doctor in the clinic. They couldn't actually understand what it meant, basically, and how to use it in the clinic. And that we have addressed by having a team of highly trained geneticists and consulting clinicians that actually write these reports. But then we have the interpretation part, and this is really, in order to, of course, be more profitable, typically you just scale up your business, it's in the volumes where you get the revenue. But we have seen that this is difficult we just have to add as many people as we are getting new samples, so in a sense our costs don't go down. And the bottleneck is really in this interpretation process, which really requires a lot of manual and brain work, and you have to have skilled people performing it. And that's where we are now implementing more automation, and we have started our first steps into AI, to see how that can help us. So the solution that we are trying to build is something we call Clinical Interpretation Assistant, or CLINT for short. What I'm showing here is essentially how the interface look like for the geneticists that use this. They have a tool on the left side there, essentially, that enforces a specific workflow. So there is a sequence of steps they need to take, and they have to perform certain decisions based on input data that they are looking. And their input is mainly in like a big table. So every row here represents a defect in the DNA or something that is different from what we expect to see there. And then all sorts of annotation information that our pipeline is gathering from a variety of open source databases, our own database. There are softwares that try to predict what a specific change in the DNA could cause in the function of the protein that this gene is coding for. So we are providing all this information, but still it requires a human brain to look at that, combine it with information that we are visualizing there on the right side from medical literature and from visualizations that we can do of the data, basically. And ultimately, they have to classify each of these rows that we have in the table, whether or not it can explain the disease. So we call this that it's pathogenic or likely pathogenic. And vice versa, they also need to start to build our knowledge database. So it's also important for us to know the ones that are definitely not linked to the disease. So we can store this information, and the next time we see this, we are able to immediately remove it from further consideration. Okay. And if we look at what is the process that is happening in this software, what they're trying to achieve, as I was mentioning early on, the sequencing instruments, they are actually outputting terabytes of data every time they conclude one of these analyses. Obviously, that's overwhelming for any human being to handle. So we first have our bioinformatics pipeline that does the number crunching, but even after that, the data that we're talking about is in millions of pieces of information, so still way too large. So the first thing that happens in this software is to try to just filter away based on some quite easy criteria. So these are fixed cutoffs for certain particular metrics that we are accumulating that just removes a lot of these rows in the table. And then the geneticists are left with something between 100 and a couple of 100 rows that they have to actually assess whether or not they can be linked. And then they can start in what we have in the middle box here, the classification process. And what we have developed is a schema that they follow that helps them to assess, is it this category or that category or that category? And that works fine with them. It ensures that we get a systematic result. No matter who is looking at it does the same thing, so we get the same end result. But the problem, again, is it's actually based on fixed cutoffs, not necessarily all the information that is underlying this data. And then in the end, when they reach the list of variants, genetic variants that they believe are associated to this, they start to write the clinical report. Okay, so we have kind of automated the first part, the filtering part there, and now we are about to start the journey on how to sort of fix the second step. And we have also done some baby steps into helping them in writing the report. So it's automated to some extent. And I can show a little bit of these things. So first, one piece of information that we have to, or that they normally would have to look up for themselves by Google searches or going to medical literature databases, and then inserting information regarding a variant. For example, the name of the gene where this variant occurs, the name of the disease, and then see what has other people found out here. But that's very time consuming. So we are using an IBM developed package here from the Watson Explorer solution to do some more clever text analytics. So this is able to now scan through 30 millions of medical scientific papers and identify and bring up the ones that are most relevant with a variety of criteria. So this is sort of artificial intelligence already, but it's not as clever still as we would like it to be. But this exists already today in our solution, but it needs further development. And I don't know if we're going to continue with the Watson Explorer or other tools for doing this. Now, another thing that we have just sort of done a small pilot study to assess the feasibility of this is that we, instead of a geneticist doing this classification, so realizing is this genetic variant, does it belong to the category pathogenic, likely pathogenic, or it has nothing to do with anything. Rather than having them do that, we would like a computer to do that and deliver some sort of metric on how confident it is in that. And when it's not confident, obviously, then the human brain has to take over and deal with that. But obviously, if we are able to do this in, let's say, a majority, maybe 80% of cases, we are really reducing their workload by 4 times. So obviously, then that will help in our scalability problem here. So we have a few criteria for this classifier. Obviously, it has to be accurate. But one thing that is very important for us, because we are providing information that ultimately can affect a patient's treatment or progression of the disease, we are accountable for everything that we do. All the decisions made in the lab, we have to be able to prove why we did it in a certain way and what was the underlying data. So having a black box classifier solution, like most deep learning technologies, for example, where you're employing something like neural network with many hidden layers, you might be able to arrive at a very accurate classification, but it's difficult to explain how you arrived there in a way that a human being can look at this evidence chain and say, yeah, it seems reliable, I believe in this. So that's a challenge. We have to have an interpretable sort of classification method. And of course, otherwise, criteria that normally applies, it has to be robust to outliers, and that's especially important in our case where some of the data points are really out of, I mean, we don't have control over the quality of all the data that we feed it. and therefore there will be outlier data points that could sort of lead it off track. So it has to be robust to that, and of course not being necessarily overly prone to overfitting. So methods that we have been looking at least so far, that could do this is some sort of decision tree where it's easy to afterwards extract how it arrived at a certain conclusion. The problem with those is that they are normally pretty prone to overfitting. But then there are extensions to the decision trees, for example, the random forest method, which tries to mitigate this by including a degree of bootstrapping or bagging technology. I will show a slide to explain that. And then I'm just outlining how we plan to do the training of this. So, of course, the only way or the right answers to the classifier that we have, that is the cases that we have analyzed so far. And geneticists have made these decisions. So we have to trust that that is correct. And that is what we're going to use to train the model. And then we will validate it on an independent set ultimately. So half the data we will be using for training and the other half for validating these things. And then it's a matter of implementation. Now we are into technical details, but obviously there are many libraries offering these kind of algorithms. But in our case, it would have to be a pretty fast one because we hope this to happen in real time as the geneticists work with the data, and there shouldn't be any delay. It should just spit it out like that. So one consideration we have is using packages available in the Julia statistical programming language. But it's not for sure that that's the final solution. So this is just a schema of how the random forest works. And as I was telling you, essentially, as you can see there in the middle, it's a bunch of decision trees that it does. But the cool thing about it is that it does a lot of them where you can tell it to randomly remove one or more of the input variables and see what classification you achieve that way. And ultimately, you can then use the consensus score or the majority call at the end. And that way, you can remove, obviously, the sensitivity to outliers. And in addition to that, you will be able to calculate some sort of p-value or confidence score on the classification itself, which is also important for us. So it can guide the geneticists to when do they need to actually take over and when can they trust the system. Okay, another thing we have realized now is that many of the databases to which we sort of hook up and extract information from, they have a lot of flaws. And the major flaw being that actually it's not case-based. So they haven't put in information about individuals that have a disease, but they are actually variant-specific or specific for a gene or specific for something else. And we would like to build up something which is case-specific, which would allow us to actually present information to the algorithm or to the geneticist in a more useful way. And this is actually quite a big project, and I don't have a solution for how exactly this is going to happen. But what we have in the middle there, the input, that is, again, all the medical literature. And these are really scientific journals that are written with human, normal, natural language, obviously, and they can contain some of the information in the text, in the normal sentences, a lot of information in tables or in figures, and we actually need to extract the useful stuff wherever it is from in these journals. And again, we have about 30 million of these. Many of them are actually using so old technologies that we probably wouldn't trust them. But in any case, that's the input. And what we can define is what constitutes a case. So we would be able to recognize patterns in this text. We can make definitions of how is variant information conveyed normally, what kind of syntax is used. We have control vocabularies for the disease names, disease definitions, and the genes. They are for sure a known entity that we can provide. And then we would have to come up with what sort of associations are important. So when you're scanning through sentences and you see the gene name, the particular variant you describe, and you see some case information, what kind of connecting words are important to have at the same time. And I guess there is no no limit to how sophisticated this can become or needs to become. We don't really know that at this point. But our goal is to then extract this information and build up case-specific or case-centric relational database of information that will make our classifier more accurate and will provide information in a way that makes more sense to us. And I mentioned in the beginning also this report compilation that is needed as the last step. And today this is semi-automated. So based on the variants that these geneticists have selected to be pathogenic or likely pathogenic, the software is able to extract background information that we have pre-compiled. So humans have been actually writing these pieces of text describing the genes, describing some cases that are known in the literature and so forth. But this is very static. So we did this effort once, and it's very time consuming and resource consuming. So it's really difficult to keep it going, and actually every day there are tens of thousands of medical articles being published, so it's impossible to keep up in a manual way. So we would need the system, first of all, to be able to update itself, extract the information that we were first putting into the database manually, if it can do that automatically, and also realize there is new information published, so I need to update this part. So a lot of text mining and crunching. Okay, so that was just giving you some ideas of the stuff we do and the challenges we face. And some things we have started to solve, some things we don't really know how to solve yet. But we believe that these are necessary in order for us to go all the way to making now this genetic testing completely a mainstream technology that is going to be routine when you go and visit the doctor with some problem. So thanks for listening to that. But before ending, I would also like to just take the opportunity and say that we are looking for people to join us in these efforts that I was outlining, software developers in general, but now because we have these very specific projects where a lot of AI is needed. If any of you guys here today is interested, if you know somebody that might be interested, please forward them to me. You have the email address there. On our webpage, we have information about these positions that we're looking for. And it will continue. As it looks now, we are just going to be expanding and expanding. So the rest of the year and into next year, we will be looking for more people. So thank you for listening.
When you have the genetic data and then you combine all the data from the papers, there's lots of information beyond the genetics, and which are the proteome and other. So what is your kind of so what do you actually include in the models and what you don't? So it's not.
Yeah, at the moment, at least the data that we're collecting is, it includes also from protein level, meaning, for example, when you try to estimate the consequence a genetic change has, you have to take into account what the protein is doing that the gene is coding for. And that information is output from a variety of algorithms that tries to tell you whether or not the functionality of this protein is first of all important in this disease case, and then is it likely to be disrupted by this particular change in nucleotide. So it's not only genetic data, we have things like that, and obviously it's important then also to know about the case itself. So descriptions, they are made in terms of treatment, or if they should be avoiding certain combinations of drugs when they get the treatment, or other indications. And that has to be extracted from this literature in some kind of intelligent way, obviously.
So one area where deep learning has been successful is in exactly on predicting the slicing, because the protein that is coming from the same gene does not necessarily have to be different, kind of similar. It changes when the RNA is sliced. So do you have any kind of let's say ambition to look at these topics as well?
Yeah, I think that several of the algorithms that are predicting again how should we say, the severity of the consequence that the nucleotide change is having are incorporating this type of information that you're referring to. And I think that we don't have ambition ourselves to build these tools that do these particular things. So there are research groups specializing in all these areas. So we are just looking into how do we incorporate that in our models. But for sure, we have, I didn't show you the full table of information that we now give geneticists and feed the algorithm, but it's more than 100 columns of either numerical, categorical, or other types of data. And it will just grow. Okay, one more there.
You mentioned that you prefer decision tree types of classification so that you can find out more about the decisions and all that. But would it be enough for you to just know that how confident the network is of this, or do you really need more understanding of that? Because
yeah, if you ask me personally, I would trust the method. I wouldn't need anything more. But actually, we are regulated under, there are, well, several types. So if you're doing clinical testing, then there are organizations in every country where we're operating that have laws about how you should be doing things and not. And they actually have this requirement. So to us, really, at this point. And I imagine that it's still sort of new technology that at some point, you know, people will just have to start trusting these things. I mean, and we do it everywhere else. So why not here? But at the moment, we would need to have the underlying. I think this one.
Both approaches and have the other one maybe find something more.
Yeah, because it can be that the more complicated and deep learning are actually more accurate or find other patterns. So definitely while we develop this, I mean, we have to ensure ourselves that whatever we do is anyway as good as it can get also to begin with. So if we notice, of course, there is a huge accuracy difference or something like that, then yeah, let's see what we do at that point. We have just looked at a very small subset of data to basically see are we able to achieve anything close to a confidence level that we want, and it looks promising so far. But yeah, we are open to all sorts of solutions, and I was just outlining, well, basically this was the study, the feasibility study behind it that generated this type of information, but we're not really fixed on a solution.